Okay, hi everyone. My name is Erwan Grasland. I'm heading the market modeling and planning function at Lonza. Today I'm going to tell you about the work that we do in leveraging clinical data to build predictive model and better understand our market. First, a few words about Lonza. If you're not familiar with the company, we are a pharmaceutical company, but specialize into the manufacturing of pharmaceutical drugs. So we don't develop any drug. We manufacture the active ingredient on behalf of our customers, so typically biotechs or large pharma companies. And we are active in a wide range of modalities, from small molecules to mAbs, ADCs, mRNA, or cell and gene therapies. So first question, what is our market for Lonza? So you're all familiar with the clinical journey of the clinical development of pharmaceutical molecule. We start with drug discoveries, then do animal testing preclinical, and then phase 1, phase 2, phase 3 commercial. Unlike pharmaceutical company, our market is not molecules that are launched or that are addressed to patients. Our market is any molecules that require manufacturing services. And this typically happens as soon as phase 1 or even sometimes preclinical. So for us at Lonza, our market is how many molecules do we have worldwide in phase 1, in phase 2, in phase 3, in each specific modality, because whenever a molecule enters a new clinical phase, this is typically when they will reach out to us to require manufacturing services. So in order to understand our market, we need to understand how fast and how successfully our customers' molecule advance through clinical phases without access to private information or to sensitive information that is not communicated beforehand, only based on public data. So in order to do that, we have developed machine learning models to estimate the likelihood of success and duration of molecules. So all pharmaceutical molecules are not equal in terms of success of clinical trials. You see on the right the result of some analysis that we do. Sorry about the redacted. Unfortunately, we had some, the legal department tend to be a bit sensitive. But here you can see that, for example, on the first line, if the drug is developed for a sensory organ indication, so for example, deafness, so on average, this molecule are slightly more successful in phase 2. If the molecule is going through a hybrid phase 1-2 trials, the likelihood that phase 2 will be completed is slightly lower, which makes sense because we're trying to do both phase 1 and 2 at the same time. If the drug has an orphan disease indication or designation, it will be slightly more successful. And at the bottom, if the drug has been suspended in the past, then the likelihood of success is slightly lower. So we have over 50 different features that we look into. And the general idea is that each of these features is statistically correlated with a slightly higher or lower success chance, or a slightly faster or slower clinical trial. So the idea is to try to train a machine learning model on all clinical trials that have been performed the last 20 years, using all of these features so that the model can learn automatically this pattern and infer or propose a certain success rate or duration to a molecule based on what is publicly known about this molecule. With that, we can build charts like this. This is a probabilistic forecast about the future state of a single molecule. So you see that this is a molecule that was in phase 1 at the end of 2024. And you see in green, the likelihood to be in phase 1 is decreasing every semester, while the chance to be in phase 2, to have moved to phase 2, is increasing and peaking until about 3 years in the future, and then start to decrease because at this point, there is more and more chance that it will have moved to phase 3 in dark blue. And all at the same time, the chance for development to be interrupted is growing. And you can see that for that specific molecule, the likelihood to reach commercial in 10 years is just 5%. So this chart here is specific to a single molecule that we have modeled. And what we have built is a system that builds this kind of charts for every molecule that is currently in active development anywhere in the world. So there are about 20,000 such molecules that we have heard of. Now, we don't use that, or almost no one uses that directly. This is still a bit conceptual, a bit theoretical. So we have built applications on top of this. Market modeling, the market modeling function at Lonza is part of the marketing department. And so the tools that we have developed are more specifically tailored for marketing use. And here I'm showing the marketing journey of the marketing journey of molecules. So we start with first estimating what the market size. So that is what the first tool is for, Pinwheel. I will get into each of these tools after, but the idea is here we can know how big is our market. The second tool is to scale our acquisition effort. Out of this market, how many molecules should we acquire based on our objectives or capacity? And the last tool is about prioritization. Once we know how many molecules we want to acquire, which one should we select? So I like to give small names to our tools. So this is why they all have celestial object names. And I'll start with the first one, Pinwheel. So this is an illustration I like to share to show that extrapolation doesn't work very well if you want to forecast the number of molecules in clinical phases. This is the number of molecules that were active in phase 3 every year until 2016. And for simplification, I consider like every modality, but you can imagine that this would be, for example, only small molecule or only MABs. And let's assume we're in 2016, and we'd like to know in 6 years, in 2022, how big will our market be? So what we usually do is to do some extrapolation, linear, CAGR, but we would estimate it to be between 1,400 to 1,600. Now, this is what actually happened. After 2016, we had a stagnation for a few years. And the question is, why? What happened? And more importantly, could we have seen this coming? And the answer is, we could have seen it coming by looking at the number of molecules in the other phases. So here in green, you have the number of molecules in phase 1, light blue, phase 2, and again, phase 3 in dark blue. And if you look carefully, you can see that the stagnation in phase 3 can be, it looks like kind of a shift of the stagnation that we have seen a few years earlier in phase 2, which itself is coming after a few years after a stagnation in phase 1. And stagnation in phase 1 started in 2008. That was financial crisis. So I guess at this point, pharmaceutical companies were less eager to start new clinical trials. So the idea here is, if you want to know how many molecules you will have in the later phase, you shouldn't look at how many molecules were in that phase a few years before, but in how many molecules were into the previous phases before, and simulate how fast and how successfully this molecule will go to the next phase. In 2016 here, there were just not enough molecules in phase 2 to sustain the growth that we had seen so far. So it was only a matter of time until we had stagnation or slowdown that we could have anticipated. So this is where our model come into place. So as you remember earlier, I presented how we do probabilistic forecasts for every single molecule. And we can group all of the molecules, all of the forecasts we have for every molecule in a certain modality to build more accurate forecasts that will capture the change of the shift in trend that might happen because of this propagation of trends. So this is a view, for example, of the when we started using this model back in the January 2024. And you see the number of molecules that we had in phase 1, 2, and 3 in blue, red, and green. And in dotted lines, this is what our model was forecasting. So, of course, there is no like huge changes, but the model was still proposing that, suggesting that the acceleration or the strong growth that we're seeing in phase 1 would decrease a bit. In phase 2, it was relatively expect a growth that is not very different. And in phase 3, the model was proposing that or forecasting that after a year of stagnation, then the number of molecules would grow again. And this is the actual numbers. And so one good thing is that our results were quite close, but more importantly, the difference between what we would have expected from a naive projection and our own projection is the same. It's the same as what we have been seeing actually. So the actual number of molecules in phase 1 has been slowing down, and in phase 3 it has been growing again after this stagnation as we have forecasted. So by using this modeling and aggregating it at a global market level, we're able to have better prediction about what the market is going to be. Of course, this forecast here is only for a short term, but we have it up to 10 years in the future, which is very important for us because our plans are that long in the future due to the time it takes to build new manufacturing plants. Now, once we have this and we're able to estimate with better accuracy how many molecules we'll have or how big our market will be in the future, it's time to know how many of them would like to manufacture. And this is where another tool that we have developed comes into place. This tool called Suban is about portfolio forecasting. At Lonza, we have the chance to work with a lot of molecules. So I think we are working with more molecules at any given time as the combined portfolio of the, I think, 5 or 10 largest pharmaceutical company. So we are literally working with hundreds of molecules at the same time. And one key question we have as a manufacturer is, how many molecules will we have in 5 years, in 10 years? And do we need to build new assets? How much revenue will that represent? So we can use, again, the probabilistic forecast that we have built to simulate that. We start with our own portfolio and would like to see, okay, across all of this, maybe 100 molecules that we have in phase 1 in a certain modality, how many of them will be in phase 2, in phase 3, or discontinued into say, 5 years? How many will be discontinued? And if we add some more across the years, how many? What will be our typical portfolio at the end of this period? So the idea of this tool is we feed it with our own portfolio. We feed it with an assumption about, okay, let's say we plan to acquire 50 molecules in different phases per year. We feed it with some parameters. When a molecule in our portfolio reaches a new clinical phase, how many batch are we going to manufacture? How many slots of manufacturing will it take? And how much revenue will it represent? And the output is a probabilistic forecast, or an estimated forecast about what is the composition of our portfolio, but also how many batch are we going to manufacture by modality, by asset size, even by site, if we can provide this information. We can also slice the result by revenue, calculating how much revenue comes from our current portfolio versus how much would come from new acquisition, not just from the new acquisition, but from the follow-up from any molecules that we acquire. So key business application here would be forecasting how our portfolio will look like, asset management, deciding whether we need to build a new asset to handle the additional acquisition that we have, and importantly for this application for us in marketing, scaling the acquisition effort. How many molecules do we need to acquire now to reach a certain target in 5 years? Taking into account that if we acquire a phase 1 molecule now, a certain percentage of them will move to phase 2, phase 3, and create recurrent business. And now for the last question, which molecule should we target? So this is the last tool I'm going to present today called Proceeds. And the idea is to identify which molecule are the best candidate for Lonza. So I like to take an analogy for how we capture molecules. So the traditional approach that we had until recently was kind of similar to fishing rod. Our efforts are in branding and marketing campaigns so that customers hear about us and reach out to us for us to manufacture their molecules. And then we have a choice of different molecules, and we just pick what we have in a way to fill our assets. Now, this is not selective, and it might not be we might not select the best molecule. So at the end of the spectrum, we could imagine going to spearfishing. So we know which molecule are the most promising because they're in phase 1 and they are going to be blockbuster, and we know that they are going to be launched. Unfortunately, in the world of pharma, you have a lot of uncertainty. You cannot know in advance which molecule will succeed. So what we're proposing with this tool is something closer to netcasting. So the idea is to use our model to identify which group of molecule, or to show the areas where statistically molecules are more likely to be good, more likely to be successful, and provide recurrent business for future phases if we acquire them early. So this is a short overview of the tool. We derive from our model several metrics that are relevant for marketing. So what is the LOA, the likelihood of approval for the molecule? How much time will this molecule take to reach, to be launched? What is the probability that this molecule will move to a new phase into the next 12 months? Because probably that's a good moment to reach out to the customer just before the molecule moves to a new phase. We have a few more metrics that I'm not going to go into, but the idea is that we have this screening tool where commercial developers can go into and filter group molecules according to several criteria and select the one that have the most promising prospect for us as a manufacturer. Thank you for your attention, and if you have any question, glad to take them.
Any questions? Thank you so much, Erwan. Maybe I have a quick question. So you mentioned those 3 different models that you use for different kind of purposes. But do you follow a overarching framework or decision matrix which tells you when do you need to run these models for what purposes and so on?
Yeah, I would say that's something that we're working for. Up until now, we're working more into like a service provider to the different function organization, but we realize that there is no overarching process, and every platform, every business unit that we have at Lonza is kind of having a different approach. So that's kind of the next step, go beyond the pure modeling task and go into change management.
Great. And final question, as always, what role do you see AI playing in this going ahead?
Sorry, can you repeat?
What role do you see AI playing in this modeling going ahead?
Yeah, this is really an exciting topic for us because all of this modeling that we do is based on structured data. We do machine learning. We have like our database, which is like things that we pay for. And these external databases are often incomplete, and they have issues, and they are not updated like necessarily very often. We could theoretically plug to different data sources, which would complement each other, but then we run into a lot of hassle with how do we merge the data, how do we deduplicate the information. So what what we're about to investigate now in 2026 is how we can use RAGs to retrieve automatic information from company website, for example, or from clinicaltrial.gov to have the information as soon as it is published, to import it, structure it in a table, and then update our model with the latest information.